Hello guys, this is Vineet and welcome to the molecular biology series, chapter number 6. So today I'll be discussing about the kinetic classes of the nucleic acids. Okay. So in the last lecture I have discussed about the reassociation kinetics of nucleic acids, and the major findings of this reassociation kinetics are occurrence of this repetitive sequence in the nuclear genome. Okay. So there are a lot of tandem repeats or interspersed repeats in the nuclear genome, and the reassociation kinetics are much more greater for this repetitive sequence when you compare with the normal gene sequences, or unique or single copy sequences which are present in the nuclear genome. So these repetitive sequences which are present in the human genome are classified into tandem and interspersed repeats. First we'll look at the overview of the human genome organization, and then we go into the main topic, that is the repetitive sequences of the DNA. So if we look at the genome, the organization of the genome, 54% of the human genome is occupied by the repetitive sequences, that is junk DNA. This is not useful DNA, and this is entirely contains lot of unwanted sequences which are not encoded into the protein. Okay, so this kind of repetitive sequences or the junk DNA is about 54%. Okay, and whatever the actual protein coding DNA or the RNA coding DNA is there, that is only about 25%, or 25%. Okay, so this genome, human nuclear genome, is categorized into two categories. One is the genic related or the extragenic related DNA. Okay, so these are the two categories of the genome. The genic DNA contains the coding sequences and also the non-coding sequences. Okay, so coding sequences mainly involve the genes which code for the enzymes or the proteins, and non-coding sequences or the non-coding DNA sequences mainly involved in the transcription of RNAs, ribosomal RNAs, mRNAs, hnRNAs, tRNAs, microRNAs, small nuclear RNAs, and all these things. Okay, so these RNAs mainly come from the non-coding. Okay, whether it might be the regulatory RNA or the RNA which involves in the protein translation, all these RNAs come from the non-coding sequences. Okay, and the coding sequences majorly code for the mRNAs which give out the proteins or enzymes. Okay, so this is about the genic and related DNA sequences which comprises about 20%, 25% of the human genome, total human genome. Okay, and the rest 75% is actually divided into extragenic DNA and the unique or low copy DNA. This unique or low copy DNA is also codes for the proteins and enzymes. Okay, this extragenic DNA is further classified into tandem repeats and interspersed repeat. Okay, so this extragenic DNA is actually classified into unique or low copy DNA and repetitive DNA. So this repetitive DNA is actually categorized into tandem repeats and the interspersed repeat. Okay, so these tandem repeats generally present in specific regions of the chromosomes. Okay, so if we talk about the mini satellites or the microsatellites, they are specific to they're specific and they are located at the telomere ends. Okay, like the chromosome ends we only find these microsatellites or the mini satellites at these ends. Okay, and tandem repeats have the specific locations, while interspersed repeats, okay, so the second category of this repetitive DNA, is dispersed throughout the chromosome. Okay, throughout the genome these sequences will be present. Okay, so this is the major difference between the tandem repeats and the interspersed repeats. Okay, so this is about the overview of organism of the human genome, and remember, only 54% is what the junk DNA or the repetitive DNA, which is not at all required. I don't say required, but it is not involved in the coding of any protein or enzymes. Okay, so we only have a unique low copy sequences. So these unique or low copy sequences which are majorly genes. Okay, and we also have a coding and regulatory regions which also code for the genes. Okay, which also code for the proteins. Okay, so these unique and low copy DNA sequences code for the proteins, and these coding and regulatory regions, they also code for the RNAs which further codes for the proteins. Okay, so this is about the overview of organism of the human genome, and then we have what differences between the repetitive sequence in prokaryotes and eukaryotes. Okay, prokaryotes mainly contains the unique DNA sequences, mostly because they have limited amount of the genetic material. So that means there is no chance of having a repetitive sequence in them. But some prokaryotes tend to exhibit few repetitive sequences. Okay, few in the sense very minor amounts of the sequences are repetitive in nature, but most of them codes for some kind of protein or other thing. Okay, but in the case of eukaryotes, since since these eukaryotes are bigger organisms and their body size is uh, much bigger in size. Okay, and the cells contain a lot of unwanted DNA, so they must contain some kind of repetitive sequences. Okay, and these repetitive sequences are classified into tandem repeats and the interspersed repeat, as I've said in the previous slide. So the type of DNA present in the eukaryotes is of two types. Okay, the genic and the extragenic. Extragenic contains the low copy as well as the repetitive sequence. Okay, so in this table I have given the percentages of the different types of DNAs which are present in the nucleus. So first, protein coding genes, they are of the 1.5%. Okay, so this is what the low copy component of DNA sequences will be. Okay, so this will have the high COT values. Okay, and they will have the low reassociation kinetics. Okay, so this 1.5 percentage will talk about the protein coding genes. Okay, and then we have the introns, which are also regarded as the non-coding sequences. Okay, so these non-coding sequences comprises about 25.9%. Okay, so these protein coding genes are also considered as exons. Okay, you can also say them as exons. Okay, and these introns are about 25.9%. Okay, and lines and signs, these are the retrotransposons. They transpose to different locations on the chromosomes. Okay, by using a special enzyme called as the reverse transcriptase enzyme. Okay, I'll tell you about that. So these lines and signs are the retrotransposons. Okay, so the name for the lines is long interspersed nuclear elements, and signs is short interspersed nuclear elements. Okay, so total they comprise about 33 or 34% of the total genome. Then we have the long terminal repeat transposons, which are also called as the retrotransposons, but they have the poly-rich repeats at their terminals. Okay, so this is called as the long terminal repeats and long terminal repeat transposons. Okay, they comprise about 8.3%. And then we have the DNA transposons, like in the copia elements or p bodies in the Drosophila. So these are copia bodies in the copia bodies and p elements or p bodies, these are called as the DNA transposons. Okay, so they transpose by copy and paste copy and paste mechanism, not copy and paste, cut paste mechanism. Okay, this DNA transposition, and they comprise about 2.9%. Then we have the segmental duplication genes, which have got duplicated, and then we have the simple sequence repeats and unique sequence repeats and the heterochromatin. Okay, so all total will be around 15% of, not 15, around 20%. Okay, so these all comprise around 20% of the total genome. Okay, so the major portion is occupied by the lines and signs. Okay, so these lines and signs, they occupy about 34 to 35% of the total genome. Okay, and these are known as the retrotransposons. Okay, they transpose from one location to another location by using their reverse transcriptase enzyme. Okay, first we have what? So dispersed repetitive sequences, which are interspersed, which are also called as interspersed elements. Okay, so these are dispersed throughout the chromosome. Okay, so they are not present in specific parts. Okay, so they are actually dispersed throughout the chromosomes. Okay, and then we have the tandem repeats, which are located at the specific regions of the chromosomes. Okay, either might be then the centrosome region or the telomere regions or in the mid part of the chromosomes. Okay, so they have the specific regions. Tandemly repeated sequences are the specific regions, while the interspersed repeats, they don't have any specific location, rather they are dispersed throughout the chromosomes. Okay, so these tandem repeats, which I was talking about in the previous slide. So these tandem repeats are of different types: alpha satellites, beta satellites, satellite 1, 2, 3, mini and micro satellites. Okay, so these are the different types of tandem repeats which are found on the chromosome. Okay, found on the chromosome and the chromosomes. Okay, so first one is the alpha satellite DNA, or we can also call that as the alpha DNA. So this is the most abundant type of the satellite DNA which is present in humans. Okay, in humans it is the most abundant type of satellite DNA, and this is found in the centromere regions. Okay, so if this is the if this is the chromosome, okay, so this part is regarded as the centromere. Okay, at this region we will be seeing this alpha satellite DNA. Okay, so alpha satellite DNA has repeats of about 170 base pairs. Okay, so 170 base pair will be repeating tandemly, continuous repeats. Okay, so this kind of repeats comprise makes this alpha satellites. Okay, and these alpha satellites are mainly found in the centromere regions. Okay, so the most important points are here is the type of satellite, type of repetitive sequence, or a type of tandem repetitive sequence, and the location of the tandem repetitive sequence is quite important. Okay, and second we have the beta satellites, which has repeating units of about 68 base pairs, and these are also located at the centromeres of specific chromosomes such as 1, 9, 13, 14, 15, 21, 22 and Y chromosomes. Okay, so these chromosomes will have the beta satellites at the centromere regions. Okay, and then we have the satellite 1 and 2, 1, 2, 3. So 1 contains lot of extra base pairs, like the repeating units, they range from the 25 to 48, or they may go up to they may go from 50 to 105. Okay, so in the satellite 1, while in satellite 2 and 3, the base repeats, the repeating units will be about 5 base pairs. Okay, so for every 5 base pairs, there will be the another repeat, and these satellite 1, 2, 3 DNA sequences, they are mostly found in the heterochromatic regions of the chromosomes, okay, which are regarded as the inactive regions of the chromosomes. Okay, these heterochromatin, these regions, these are not involved in the transcription or any kind of gene regulation process. Okay, so this is called as the heterochromatin region, and in these heterochromatin region we will be seeing the satellite 1 and 2 DNA sequences. Okay, repetitive sequences. Then we have the mini satellites. So these mini satellites are also referred to as the variable number tandem repeats, VNTRs. Okay, so these are of 10 to 60 base pair repeats. Okay, and these are located mainly at the telomere ends. Okay, I have said you right in the previous slide. So these mini satellites, they are specific for telomere sites. Okay, so they are only located at the telomere ends. Okay, so if this is the chromosomes, the end parts of the chromosomes are regarded as the telomeres. Okay, and at this telomeres, we will be seeing these mini satellites. Okay, and sometimes microsatellite will be also present. Okay, and then we have the microsatellites. These are also short tandem repeats, but not very short tandem repeats. Okay, mini satellites are what short number nucleotides. Okay, so these microsatellites much more shorter than the mini satellites. Okay, so we have 1 to 6 base pair repeats. Okay, in the microsatellites. Okay, micro is very small. Okay, when you compare with the satellite 2 and satellite 3 DNA, it is much more smaller. Okay, microsatellites, and this is also referred to as the simple sequence repeats. Okay, there are a lot of techniques to identify the suspects in the forensic experiments. Okay, forensic laboratories. Okay, so they use this simple sequence repeats for their detection of the suspects in the forensic laboratories. Okay, so these microsatellites, they are dispersed throughout the chromosomes. Out of all, only microsatellites are dispersed because they have very short base pair repeats. Okay, because of that reason, these microsatellites are dispersed throughout the chromosomes. Okay, and these microsatellites were believed to be evolved from the slippage of polymerase activity. Okay, whenever the polymerase replicates the DNA. Okay, and whenever it slips out from the leading strand, okay, or the lagging strand, at that time these microsatellites were believed to be generated. Okay, so due to the slippage errors of the polymerase during the DNA replications, these microsatellites were believed to be generated, and mostly these microsatellites contains the cytosine and guanine rich sequences. Okay, if you can see in the final point. So these microsatellites were rich in the cytosine and guanine rich nucleotides. Okay, so guanine rich molecules. This is all the sequences, they contain higher numbers of the cytosines and the guanines. Okay, so in the final sequence over here, this is also a normal repeating base pairs. Okay, repeating unit. So in this repeating unit, we have 1, 2, 3, 4. Okay, so out of five bases, 4 of them are cytosine and guanine. Okay. Due to this occurrence, cytosine and guanine rich sequences, these microsatellites will allow the DNA to form triple helix and the G coordinates, okay, or tetraplex DNAs, okay, or or make the DNA to undergo into loop conformations, or loop stem formations, okay, so stem loop formations, okay. So these kind of tertiary levels, tertiary structures of DNA will result from the uh, microsatellites. Okay, so microsatellites contains the lot of guanine and cytosine rich DNA, guanine and cytosine rich sequences, and these sequences will make the DNA to fold into tertiary level, that is the triplex DNA strand or tetraplex DNA strand, or to fold into stem loop. Okay, so lot of unusual conformation will be seen at this microsatellite level regions. Okay, so this also induces some kind of superhelical stress to the DNA. Okay, so and which is of course the topoisomerase will relieve the stress. Okay, but microsatellites were most important DNA sequences which produces that kind of superhelical stress on the chromosomes. Okay, so this is what about the tandemly repeated sequences. Okay, so remember the tandemly repeated sequences will be present at specific locations except this microsatellites because of presence of short repeats. Okay, so this is dispersed throughout the chromosomes. Okay, sometimes this is also considered as the interspersed repeat of DNA. Okay, and then the second category of this repetitive DNA sequence is the interspersed repeated DNA or the dispersed repeated DNA, and this class mainly contains the transposons as well as the lines and signs, which are also called as the retrotransposons. Okay, first, we will discuss about the lines and signs, which are regarded as the retrotransposons. Okay, and then we'll go for the transposons in a special video. Okay, special lecture. Okay. So now these sequences, as I've said you, they are dispersed throughout the genome. Okay, so these are categorized into uh, two types. Okay, one is the lines and one is the signs. Okay, so lines. The name of the lines is long interspersed elements or nuclear elements. They contain 1000 to 7000 base pairs per unit. Okay, so the repeating unit will contain 1000 to 7000 base pairs. Okay, because of such huge base pairs, these are regarded as the long interspersed nuclear elements. Okay, while the signs, these signs contains only 100 to 400 base pairs per repeating unit. Okay, so repeating unit contains only 100 to 400 base pairs long, and these because of the short sequences, these are these are considered as the short interspersed nuclear elements, and these lines are uh, known as the autonomous retrotransposons, which can automatically, or which can transpose from one location to another location by themselves. Okay, they don't require any mediator molecule for their transposition. They don't no, translate their own protein, their own enzymes. Okay, and using those enzymes, they transpose from one location to another locations. Okay, but when you look at the signs, these signs are non-autonomous. That means they are dependent on the some other mediator molecule. So here the mediator will be the lines, and whatever the enzymes that are encoded by the lines, those enzymes will be involved in the transposition of this sign elements. Okay, so signs require the lines for their transposition, while lines are autonomously transposing elements. Okay, so lines don't require any kind of mediator molecule for their transposition, while signs require the lines for their transposition. Okay, this is what the major difference between the signs and lines. Okay, and a lot of college level entrance exams also have also asked these questions in their uh, tests. Okay, now all eukaryotic organisms have these sequences, lines and signs. Okay, major Drosophila and the birds also have the lines, like long interspersed nuclear elements are majorly present in this Drosophila and birds. Okay, and human frogs, they also contains lines, but mostly the short interspersed nuclear elements. Okay, so when we talk about the lines, I've said you this is a autonomously transposing element, retrotransposing element. Okay, so what happens during the initial stages, whenever an RNA polymerase transcribe this line sequence. Okay, it produces an mRNA which contains the sequence for a protein Okay, so this protein has a RNA binding property, and it also encodes a second protein which has endonuclease property as well as a reverse transcriptase property. Okay, so the protein which has RNA binding property comes from the open ending frame from. Okay, so this open ending frame is involved in the coding of this RNA binding protein. So this ORF1 protein, and the ORF2 open ending frame 2 is involved in the uh, for formation of this second protein which has endonuclease activity as well as the reverse transcriptase activity. Okay? So when all the mRNA forms gets transcribed, okay, inside the nucleus, okay, it will come out of the so when all the mRNA is transcribed and stays in the nucleus, okay, so it won't come out of the nucleus, okay, it will stay in the nucleus, and this mRNA, by utilizing this reverse transcriptase activity, converts itself into the DNA. So RNA will get converted into DNA. Okay, so this conversion of RNA into DNA will be only done by the special enzyme called as the reverse transcriptase, and this reverse transcriptase will make the RNA to undergo get converted into the DNA. Okay, and this DNA will now get incorporated into some other location of the chromosomes by using these endonuclease enzymes. Okay, what this endonuclease enzyme do is it will go and cut the DNA's. It will go and cut the sequence of chromosome. Okay, like if this is the actual chromosomes, okay, this endonuclease somewhere cuts, or somewhere on a specific region, it makes the codon chromosome. Okay, so it will cut the DNA sequence, a specific DNA sequence, or random DNA sequence, and makes the DNA to fit into this sequence. Okay, so it will fit the transcribed DNA, reversely transcribed DNA into this sequence. Okay, so these lines which are retrotransposons encode for the endonuclease and reverse transcriptases. Endonucleases, they cut the DNA sequences and fits the DNA, makes the DNA to fit into the those cut sites. Okay, and reverse transcriptase will convert the newly formed RNA to DNA form, so into DNA. Okay, so this is what the function of the lines. Okay, so this is how they increase the amount of the DNA or the quantity of the DNA in the nuclear genome. Okay, so and that is how these lines and signs comprises about 34% of the total genome. Okay, so these are about the lines. Okay, and the full length line 1 family, we have a simple line 1 family, and remember this point also, the mammalian diploid genomes have about 500,000 copies of line 1 family. Okay, and these line 1 family members are about 6 to 7 kilo base pair long. That means 6,000 to 7,000 base pairs long. Okay, and all those most are the truncated elements of about 1 to 2 kilo base pair. That means 100 to 1000 to 2000 kilo base pairs. Okay, so that means they are the truncated version. They are the broken versions of this 6 to 7 kilo base pairs long molecules. Okay, so these line 1 families are dispersed throughout the chromosomes, and by utilizing these kind of enzymes, endonuclease and the reverse transcriptase, they move to different locations. Okay, this is what the information about the lines. Okay, so then we have the signs which are also called as the short interspersed nuclear elements. Okay, so one major example for the short interspersed nuclear element is the ALU element. Okay, which is a major enzyme. ALU is a major restriction enzyme which is studied in the bacteria. Okay, so this sign family, it contains nearly 200 to 300 base pair sequences per repeat. Okay, and it makes nearly 9% of the this sign element alone makes the 9% of the total haploid DNA. Okay, and this ALU repeat is located every 5,000 base pairs in the genome. Okay, for every 5,000 5,000 base pair sequences, this ALU repeat will be present. Okay, you can find the ALU repeat for every 5,000 base pair repeats, and this signs is transcribed by the RNA polymerase 3, while the lines are transcribed by the RNA polymerase 2. Okay, so you will get the line mRNA by RNA polymerase 2, while you get the sign mRNA by the action of RNA polymerase 3. Because this is also the major difference between the lines and signs, apart from the bases and the atomos retrotransposition property. Okay, So these signs are non-autonomous retrotransposons. That means they require some kind of mediator. That mediator is what? Lines. Okay. So see this point. The signs are transposons, but they do not encode any the enzymes that they need for the movement. Okay. So for the transposition movement, they require the enzymes, but they don't encode for the enzymes. Okay. So they can move, however, if those enzymes are supplied by the active line transposons. Okay. So these line transposons, whatever the enzymes encoded by these line transposons are utilized by the sign transposons, and by using those enzymes, they move from the, or they transpose from the one location to the different locations on the chromosomes. Okay. And there is a B1 element in frogs, sorry, in mouse. Okay. So the mouse contains about 50,000 copies of the ALU related B1 element. Okay. So this B1 element is actually a member of this ALU elements. Okay. And this mouse actually contains the 50,000 copies of that ALU related B1 element. Okay. And the human ALU element possesses 2 tandem repeats of this ancestral 130 base pair B1 sequence plus an extra unreaded 31 base pair insertion. Okay. Like the humans also contain the same B1 sequence element, but it is only limited to the 130 base pairs. Okay. And we also have an extra 31 base pair a B1 element. Okay. But we don't know its origin. Okay. So we don't know where it has came from. Okay. So but this insertion of obscure origin. Okay. So we don't know about the origin. Okay. But we have the same B1 element which has been observed in the mouse. Okay. The same B1 element is present in the humans. Okay. So which is of 130 base pairs. Okay. An extra 31 base pair B1 element is also present. Okay. And when you compare this B1 element in the mouse and humans, okay, this B1 element which is a sign. Okay. So mouse has less than 100,000 species of the B1 element, so it would be classified as a moderately repetitive DNA, while humans have more than 100,000 copies of the ALU element, so it would, therefore it is classified as the highly repetitive DNA. Okay. So that means mouse has only a limited number copies of the B1 element. Okay. So the signs number of sign copies were limited. Okay. Only 100,000 copies were there. But if you look at the human genome, the sign copies or the ALU elements or the B1 elements, they are more than 100 copies. Okay. They are present more than the actual amounts. Okay. So these more amounts are actually considered as what? Highly repetitive DNA sequences. Okay. So these signs are also regarded as the highly repetitive DNA sequences, majorly as the interspersed sequences. Okay. So this is about the lines and signs, and in the next lecture I'll be discussing about the transposons. Okay. DNA transposons and also the some extra retrotransposons. Okay. Long terminal repeats also. Okay.